Okay, I think we'll get started now. So good afternoon, everyone. Welcome to the Clinical Trials Methodology course webinar series. Today's webinar on common data elements is our last in the 2018 series. Before I introduce our presenters, I'd like to remind you that we'll take questions at the end. So if you have a question, please use the chat box to the right of your screen, or you can simply unmute yourself. Also, at the end of the webinar today, please take a moment to evaluate the webinar. I'll provide a link at the end of the presentation. So now I'd like to introduce our presenters. First, we have Dr. Robin Conway. She is a program director in the Clinical Trials Cluster at NINDS with a background in neuromuscular disease and clinical neurophysiology. She's also an executive committee member and core faculty member for the CTMC. Her current interests include neurologic intervention studies, adult neuromuscular diseases, including ALS and neuropathies. Next, we have Dr. Carolina Mendoza-Puccini. She is a scientific program specialist in the Division of Clinical Research at NIH. She is the program officer for the Clinical Trials Readiness Network and the Strategies to Innovate Emergency Care Clinical Trials Network. In addition, Dr. Mendoza-Puccini manages the NINDS Common Data Elements Project. Her main interests are common data elements, clinical trials, diversity in the workplace, big data, and data sharing. And lastly, we have Alex Sherman, who is the director of the Center for Innovation in Bioinformatics at the Neurological Clinical Research Institute at Massachusetts General Hospital and a principal associate in neurology at Harvard Medical School. The focus of his work and research is conception, design, development of technology, platforms, and infrastructure for collaborative clinical research and optimization of clinical research operations in a given disease network. Other areas of interest are creation and management of disease-specific research networks, understanding the benefits of patient-centric research, and incentives for research collaboration. So I'd like to thank all of our presenters today for their time and presentations. We'll start off with Dr. Mendoza-Puccini, and I'll share your slides in just a moment.
Hello, everyone. Today, I'm pleased to be joining Alex Sherman and Carolina Mendoza-Puccini in this very interactive webinar. In my many years of experience at NIH and NINDS, I can say that this CD project is an excellent opportunity to standardize clinical research data, as well as harmonizing efforts that are extremely important and do increase efficiency. As many of you know, this CDE contract is a major NINDS initiative with many, many meetings and iterations. Alex can attest to this as well. This project is actually like the Constitution of the United States. It keeps changing and evolving. And most recently, it's been evolving in a positive direction as Carolina Mendoza-Puccini at NINDS is now in charge and has taken the helm as the program director for the CDE. Carolina?
Joy, can you please show me the first slide? So thank you all, and I'm very excited to present my project. And next, please. So I'm going to tell you a little about what is the CDE project, or the Common Data Elements Project. So this project started about 10 years ago. Actually, NINDS was one of the pioneers developing these tools. And the project was to develop the data standards for clinical research in neuroscience. So the common data elements are content standards that can be applied to various data collection models. And as Robin mentioned before, they're very dynamic and evolve over time. But CDEs are not a database, and I will talk about that later next. So the goals of the project are to develop common definitions and standardized case report forms, or CRF, and other data collection instruments. And they also help investigators to conduct clinical research through the development of these uniform formats by which clinical data can be systematically collected, analyzed, and shared across the research community.
We want to make sure that data is comparable.
Exactly. Okay, at NINDS level, we ask our grantees, and it's in the term of awards, that they use the common data elements when they do phase 3 clinical trials and exploratory trials. So usually UO1 are required. And the clinical research projects will be accelerated, and new investigators can build consensus data elements. They can start up multi-center and international clinical research efforts, and that will be streamlined.
So if you don't use them, you need to explain why you're not utilizing the CDEs.
Exactly, but there has to be a very strong reason why not to use them. For example, will be that we haven't developed CDs for that particular disease. That's the strongest reason, exactly. So a little bit of definitions. What is a data element? So it's an information that describes a piece of data to be collected in a study. And a common data element, or CD, is a data element that is common to multiple data sets that are going to be used across different studies. And these are definitions from the National Library of Medicine. And a CD is going to include a name, a precise definition, a semantic value, with clear definitions and permissible values. And they're consistent and harmonized across different data sets. Next. So the way we define the CDs and the way we ask the grantees to use them is the following. We have a general core that is going to be used across all diseases. They're general CDs like demographics, medical history, etc. And they're going to be required for all NINDS funded studies, the ones that I mentioned before. Then the next layer is a core, that they're CDs specific to that particular disease that are also going to be required. Then the supplemental highly recommended, that are important tools that the expert decided that will be great to have them in the study, however, they're not required. Then the supplemental, that they're less important in the way they categorize them, the experts, and the investigators can choose and pick what instruments they want to use. And the exploratory, they're just tools that are not validated for that disease, or they're not validated at all. However, they may be important to include them in a study. Next. We currently have over 20 disease areas. So there are about 18,000 CDEs and 1,400 instruments in total. So we have cerebral palsy, Chiari-1, epilepsy, headache, mitral disease. In movement disorders, we have Parkinson's and Huntington's, MS, ME/CFS. In the neuromuscular diseases, we have ALS, Friedreich's ataxia. In the muscular dystrophies, we have Duchenne, FSH. We also have myasthenia, spinal muscular atrophy, and then spinal cord injury. In stroke, we have other subcategories. TBI, we have sport-related concussion, and biomechanical devices in TBI. And some of them have pediatric CDEs also. Next. So the development process, this is very quick. So we initially identify CDEs, develop and vet them by a group of experts from that scientific community. Then we have collaborative involvement from other NIH institutes, other federal agencies like National Library of Medicine, CDC, DOD, etc. We also have collaboration with industry and patient advocacy groups. Whenever possible, we're aligned with other international data standards, and Alex will talk about that later, like CDES or ISO. And NINDS, our role is to provide continued guidance, review, and oversight in general of the project. Next. So these are the development steps. I'm not going to go through them, but in a nutshell, it takes about up to 18 months to develop CD. So it's not an easy process. It takes many meetings, as Roy mentioned before, a lot of time from the experts, which they do that in a volunteer basis. And then that's the timeline requires a lot of steps.
So initially we had these long CDE processes, and then we decided to go back and refine the core elements to pare it down, to make it even more efficient. So now we're going through essentially another period of growth.
Mm-hmm. And we revise every year all the CDs, all the diseases, and every year we try to develop a new one. Next. So These are potential benefits of CDs, and I can attest to that. I use them in a study. They work, and I will tell you some of the benefits. So, and I think the most important one is going to reduce time and cost to develop the data collection tools. You're going to increase efficiency. You don't have to go and reinvent the wheel creating case report forms because they're already in the website, and you can download them into several formats like PDF, CSV, Excel, etc. It's going to also reduce your startup time and the cost of your overall trial. You will not have to, and this is based on my personal experience, it will reduce the time of the meetings and aggravation of herding a bunch of people to decide what to add to your case report form. It's going to improve your data quality and facilitate data collection because you're going to use it across the study. You're going to collect your data in a uniform way, in a standardized manner, and also it's going to make your study NINDS and NIH compliant. The CDEs are not only used or developed by NINDS. There are many institutes that are also using them or in the process of developing them. They're also going to facilitate data sharing because the data set will be harmonized. And so it will reduce the time that you have to harmonize all your variables and go through all the steps to make your data set analyzable. They're going to improve opportunities for validation of exploratory elements or instruments. If you have an instrument that you want to validate for a new purpose, a new disease, or a brand new instrument, that will be an opportunity maybe to use it in clinical trial for clinical trials readiness. And then it's going to also increase the CDE awareness in the neuroscience community through publications, presentations, and other ways to disseminate the usability in the neuroscience studies. Next.
One more thing, I was hoping we'd involve more young investigators in this process. And we're hoping, and we can talk during the panel at the end, about ways to bring more ideas in and younger investigators, newer investigators.
And honestly, I wish I knew before I used the CDs for the first time in a stroke study that there were so many diseases, but it would have saved me a lot of time. And this is the website. Joy will post the slides. So the website, I'm happy to report that the current website is not as friendly as we wanted to. It was developed years ago. So it was time for us to work on a new website, and hopefully it will be up and running early next year. The contact information of our contractor, who they manage all the administrative part, my email and Robin's email. Feel free to email us or email me anytime for any questions that you have. And now I'm going to hand it to Alex, who is our next presenter.
Thank you. Joy, may we try again to share my screen because it seems that it should work now. Does it?
No, I don't see it.
One sec, I apologize. I say share screen. Screen 2, share.
Oh, there it is. I see it.
All right.
Now we can see.
Looks good.
Great. So thank you so much for joining this webinar. And what I will try to do is to try to give you more of a practical kind of advice on how to use CDs and why to use CDs. And the reason I'm kind of sort of comfortable doing this is one of the hats I'm wearing is I am running a clinical data core for multiple disease-specific research networks in neurological or CNS space, including ALS, Parkinson's, Alzheimer's, and smaller diseases as well, and stroke as well, actually. So what I will do, I will go through this. Please interrupt if you want me to, and Joy will let us know when I should be responding to some questions, but we can leave till the end. I will try to go through very in a fast manner, but don't hesitate to stop me. I will reiterate what just Carolina and Robin said. So why to use and why to harmonize? Definitely one of the major things is that when you have a clinical trial protocol, clinical study protocol, you want to start it as soon as possible. And if you start designing CRFs or case report forms and from scratch, it takes a lot of time and effort. And most likely will not be able to share your data with others because it will not be as harmonizable as we would love it to be. So certainly NINDS clinical data elements are useful, at least to understand, download, and understand what the communities are thinking about what should be used. Most likely, though, that as Carolina mentioned, so there are only 20 a lot of diseases, 20 diseases, but there are about 2,000 neurological diseases out there. So if you do not find your forms there or CDEs there, just think about more generic approach and still analyze those forms and see what and how it is recommended to approach the CDE design. So, and then another very interesting thing is that we do want to harmonize it. I will be talking about this sharing collaborations and in a little bit, but the idea is that if you're a researcher in a certain disease, you want your disease to be clinical trials ready. And for this, you have to put together as a community, do research in this particular disease. You would love to and have to put common data elements and CRFs and common approaches for meta-analysis, for biomarker and outcome measures development, and eventually you would love to aggregate enough data to attract sponsors and pharma, because as we know, most likely it's pharma who will be developing drugs for these particular diseases. So, and the last and not least, so we will prosper and our careers are dependent on publications, presentations, and so on. And certainly if you could merge data from multiple studies, either yours or data from multiple studies in the community, it will be much more powerful for you to publish, analyze, present, and disseminate. Another thing is a couple of thoughts that if you're going after proprietary standards, it will be less effective for you to harmonize and merge data. And I will show you a couple of examples on this. And then definitely another important thing that if and when we're doing clinical research and we have some findings, it would be unbelievably useful if we could start using such findings in our clinical practices as well. And certainly you want to be aware what is being done in clinical practices and how we could use data in clinical or from clinical practices and clinical visits in clinical research and vice versa. So, and certainly think internationally for interoperability, and we can talk about this as well. And then one of the important things is when you're doing any small project of your own, even a single side project, you have to start thinking, okay, so if someone approaches me and wants to collaborate, so how do I share my data? Could I share my data and so on? So, and we could talk about this later as well, but it is immensely important when you start thinking about clinical research projects to understand what should be put into informed consent forms. What should you tell your patients or research participants on how you're planning to share your data? And for example, in some of the networks that we're running, especially and I will be showing this example, a clinical trial network, clinical research network in ALS, Lou Gehrig's disease. So it's important to say that we will be sharing your data for any medical research purpose. In this case, you will not be under any constraint of how you will be sharing or with whom you will be sharing the data. Then we can talk about some consortia that recommend you on formats for data sharing, harmonization, and we can talk about the reasons why to join this formats and standards. So, where could we use the CDES? First of all, let's go from the very top. So, if we're running a formal clinical trial that should be data sets shall be delivered to the FDA for registration purposes. So certainly there are multiple standards that you have to be aware, and it's not the topic of this seminar, but if we have time, we can talk about this as well. But there are things like 21 CFR Part 11 that regulates from the FDA's or government perspective how we can or shall treat systems for clinical data capture and electronic signatures. There are certainly good clinical practices, so GCP, and that regulates from the international harmonization perspective whether and how we can trust systems that capture data in clinical research and clinical trials. And there are certain formats including so-called CDES, that we can discuss it a little bit later as well. And this CDES format actually currently the FDA requires the data to be submitted in such format. So then there are investigator-initiated clinical trials and even single-site trials as well. And if you could think about it, so even if you're not required to do it now, but if you ever think that your data that you're aggregating in this clinical trial may be used in the future as the basis for the future, phase 2, phase 3 clinical trial design, you should be thinking about similar standards that I mentioned for the registration trials, including 21 CFR Part 11 and GCP. I'm saying perhaps CDES because CDES is a format, so eventually it could be any data could be transferred into CDES format for the FDA consumption later. And then if you're going for a validation of new biomarkers and outcome measures that will be used or may be used in the future for clinical trials purposes, definitely want to make sure that what the measures that you're validating against are compliant and standardized. And then there are observational studies. And this is what Carolina mentioned before. So certainly if you're developing your clinical disease space and domain, so you want to be considering sharing and future meta-analysis. And it takes enormous effort if you have not thought about standardization and harmonization before you start developing your common data elements and case report forms for data collections. And then we'll be talking about this as well. There is a current trend, very important though, coming from the FDA. So in smaller diseases, and especially in single gene diseases, where more and more open label clinical trials we can see. So the FDA, and I will show you on the next slides, the FDA encourages collecting and sharing natural history data in such diseases to be used as control arms against the open label data acquired in the formal clinical trial. So, and these are the recent messages coming from the FDA itself, and they're talking about rare and ultra-rare diseases, but they're talking about clinical outcomes and patient experiences. And we could talk about this later as well. But one of the major things that, if you could look at the second paragraph, that eventually use natural history models to augment the need for placebo arms in studies of drugs. And quite recently, there was an announcement of a small study in SMA. You can see here highlighted that the vast majority of patients demonstrated improvements superior to the natural history, right? So, but the idea behind it is that this study or this data has to be at par and trustworthy as if it were collected as part of the regulated clinical trial. All right, let's step back for a sec and let's try to figure out what data do exist in clinical research kind of continuum, and let's think about how we can use the common data elements for harmonization, sharing, and collaboration. So in general, the dream of any research or any clinical research is how to make each and every patient to turn into a research participant. And in most of the diseases, it's 1 to 3% of patients in clinical research. In rare neurological diseases, it's coming to probably 15 to 20%, but certainly it's not enough. So let's look at what we can have in terms of the data. So we're talking about clinical trials data, if there are clinical trials, but then there are health records, natural histories, bioimage banks, patient-reported outcomes, PROs, genetics, omics, cell lines, mobile apps lately, and so on. So, and I will be talking about, as an example, certainly not as a promotion, several tools that my organization developed and utilizes for such data capture and sharing. So, first of all, we're trying to look into this sort of easier tools. So we're always looking for more data. And one thing we did is we went after clinical trials data in ALS and decided, wait a second, so companies collected a lot of data, why cannot we use it? And we went with a company organization called Prize for Life. We went and asked companies to donate data from their clinical trials, and companies like Teva, Novartis, Regeneron, Sanofi, Biogen gave us data. We had some data sets from our NILS consortium, the largest in the world consortium of ALS clinical research sites, 127 sites in the world. We had data from our consortium and ended up with 23 clinical trials data sets that we harmonized, merged, analyzed, and there were challenges. Some data came without data dictionaries, some data came with obsolete data, obsolete outcome measures, and so on. So, but eventually we managed to put it all together, and we ended up with 11,000 patients or subject records from 23 clinical trials. And we, right now, it's the biggest reference data set in the world for anyone who wants to study ALS as the disease. So, but I just want to show you a couple of challenges. So in a very simple example, so these are demographics forms and coming from multiple studies. So first of all, some data came in French, for example. Some data came in very weird. And as you can see on the left in blue, so those are the common data elements that we use, and actually NINDS common data elements use as well. So you can go up to gender, male, female, or sex, currently male, female, right? So race, white, black, Asian, and so on. So on the right, there are several studies, and you can see that, so for the field sex, there was one option was M and F, another option was and FOM. So basically, I mean, I guess we could figure it out, but some data may have come in Japanese and so on, so you have to be aware of this. Another interesting thing that some races in multiple countries are being identified differently. So in this case, we have race Japanese and race Oriental as two separate options, or race Asian on the top study. And I mean, it's an easy guess, but what I'm trying to say that each and every field or data point in each data set, you have to put and map to your standard data structure. So this is a challenge. So certainly if we were kind of coming from the very beginning and saying, oh, let's harmonize, it will be much faster and easier. And now actually it is much better because the companies that start studies in ALS, they go to this data set and download and analyze data, and they're aware of such challenges. Because they're claiming, at least, and some of them donating data to this resource, after they finish their clinical trials. So in this case, it is much faster for us and easier to map and upload data. All right. So, and I don't want to spend too time on here, but one of the important things to understand how we could use this data is that we created the crowdsourcing challenge, where people were downloading these data sets, and we have about 1,100 solvers downloading data, and we announced 3 winners instead of 1, and none of the winners had anything to do with ALS nor with medicine. So this is very important to understand, that there are a lot of people out there who have some knowledge on how to analyze data, and they have no access to data. And this is one of the tools to kind of bring wisdom of the crowds to clinical research. So you could look at this later, and there is a paper in Nature of 2015. But more and more, besides clinical trials data, we can talk about, and there are talks about, rather, so-called real-world data. So basically, just to understand what it is, the easiest way to understand is anything but regulated clinical trials data are considered real-world data, meaning data from health records, data from claims, data from cell, mobile phone apps, and so on. These are all considered real-world data. And there are multiple tools that could be used, but one of the challenges, actually, especially in neurology, and especially in neurological rare diseases, is how do we identify patients? So, as you know, when patients are coming into our clinical trials, you assign subject IDs to these patients and aggregate data information based on those subject IDs. When the same person comes to you again later or elsewhere, a new subject ID is being assigned, and data are collected based on that subject ID. Basically, by design, there's no way that we can merge data for the same person from multiple studies. So what we came up with actually NIH help is with the tool that is we call Neurological Global Unique Identifier, because most of our research are in neurology space, but basically it could be any global unique identifier. And this technology was initially developed by the NIH. We improved it a little bit, but just for your understanding, there are multiple good or global unique identifier servers that are being run around the world, actually. And NIH, my understanding or recollection, runs 5 or 6 of those that are disease specific. So we run our own server, and which is harmonizable at the back end with the NIH, but we developed many more tools to be used with this app. So you could look at this later, but basically you enter information on the patient twice to avoid any confusion. Then you say generate GUID, and basically what happens is that this server, the client side of the server, encrypts and packages this information into 3 so-called hashes or gibberish strings for those non-techies. And then it sends this gibberish strings to the server. And server, based on those strings, generates a random number or random character string that is called GUID or GUID. So basically, this string does not contain any patient information. And every time you enter the same information onto the client, so the same string is being returned. Basically, the server says, oh, I already have these gibberish strings, and returns this global unique identifiers. So, and another thing that we did based on this propagation of neurological global unique identifiers, we created a brand new platform, brand new in terms of the concept, how it's being used. So most of the clinical research data capture systems are being used as siloed kind of clinical trial or clinical research project centered systems, where you create a clinical research project, you aggregate data based on subject ID, done, next project. Here we're creating, we have a platform that we call NeuroBANK. So you're creating a person, a patient, with this neurological global unique identifier. And then you say, okay, so this person consented to this particular study or trial, then visits and forms for this study, for this patient, become visible for that study team. When the same person or patient consents to a different study, only that study team can see forms and visits for this patient. And they may not even suspect that this research participant, participates in more than their study. But if they happen to talk and collaborate and, God forbid, share data, so certain data elements may readily be available for the second or fifth study. And I will show you how we can do it with the example from ALS. So also we're running it's important to map and match and track biosamples and biorepositories around the world. And we run those biorepositories, so-called virtual biorepositories, with labels on the vials and postmortem tissues that contain the same neuroglue, neurological global unique identifiers, and links to the study IDs. And also we're talking about patient-reported outcomes that currently are very important or becoming very important lately. We can discuss this during our Q&A session. And then, so basically from example, in ALS, we're running currently 22 clinical studies and from 80 sites in 14 countries. And all 22 collect clinical data, 15 collect biofluids, including longitudinal CSF and plasma and serum and urine and so on. So 11 collect DNA samples for whole genome sequencing. 5 collect iPSCs or PBMCs for iPSC generation and cell lines, and everything is shareable. So basically, we call it ALS ecosystem, and we have currently about 4,400 people with ALS. That's a lot, actually. And about 8.5% of research participants participate in more than one study. We have one champion participating in 6. And 2,300 DNA samples are available, whole genome sequencing for sharing. Almost 16 PBMC samples were collected for cell line generation. We have 176 cases of postmortem tissue collection, and we have 76,000 vials of biofluids. And everything is readily available for anyone who asks. So this is when we're talking about collaboration sharing. This is what we have. So another thing, we're always looking for incentives. How do we bring more researchers and clinicians into clinical research space? One of the things we discovered that a lot of pain comes from inability of clinical researchers and clinicians to analyze their own patient populations, because most of health record systems do not allow them to do this. So we developed an analytical tool that we call NeuroChart, but it could be CardioChart or any chart. So, and the tool could sit on any data set, but in this particular example, it's being used in NeuroBank itself. So basically, we're developing multiple charts, and then you merge them into a dashboard. And on the left, as you can see, you can put together a lot of filters or analytical parameters that you can play with. And based on this, the data changes in real time. So we can go and look for females versus males. You can go and look for fast progressors, as you can see here in ALS for functional rating slow versus slow progressors, and you can see which drugs are in slow progressors using versus fast progressors, which are, for example, you can see here that our site of onset here is prevailing limb onset, and here it's prevailing bulbar onset, and so on. So you can also track in your network how many gene mutations or patients with gene mutations you may have. You can also see individual slopes for functional rating scales. And then when you start playing with data and you can narrow your search to a particular population, you can click on the export button and export the entire record or clinical records for these patients into Excel or CSV format for future analysis. And another thing, what's important, so we're not holding other people's data. So we do not have whole genome sequencing files or images in our data sets, but we know where these inventories are. So basically, when they're looking for certain things, including whole genome sequencing files in this case, so we can show them here that these whole genome sequencing files are located at New York Genome Center, and they can go and ask for that information based on these neurological global unique identifiers on the left. So here another example that you can go and check for disease progression of particular patients. Actually, that's another important thing to emphasize. So, a lot of times when you're doing in your clinical practice, when you see a patient in front of you, you cannot compare that patient to the rest in your clinic or to the rest in your network. And this tool allows you to do that. So we can look at individual patient, you can see vital signs and outcome measures for this patient. At the bottom, you can see the drugs or conmeds this patient is on, and you can track, for example, if this drug starts here, whether there were any changes in vital signs. But also you can say, aha, so this patient's function rating is low compared to the rest in my practice. And those are vitals compared to the rest in my practice or the rest in my network if you want to share your data with others. So individual slopes against everyone else. And also you can find what biofluids are available for this patient in your biorecords. So basically the idea behind this again, so if you could merge all this information, you can share this information, again, as I would love to emphasize, with appropriate language so for sharing and data use agreements and so on. So and I will be happy to share such language and agreements with you. It's very important to emphasize this. Without this, IRBs will not allow it, or your legal department. So, and then probably the last kind of piece of practical advice, if you don't mind me saying this. So, first of all, any CDE that you're downloading from the NIH website or elsewhere, to make sure that you understand that this is a living organism, so it's not a dogma. And tools come and go, and there are some becoming obsolete. New measures are being developed. So so view anything or everything that you download from NINDS website as kind of guidance document rather than a etched in stone document. So and then you have to understand that common data elements are not case report forms. So you can develop case report forms as you wish, but to make sure that common data elements are being not only named but also used properly. So ideally, certainly, if you use common data CRFs, it would be all the same, and because if you keep your own electronic data capture systems, in this case, it's very important to keep library. So nature of research projects also determines which common data elements to be used. So, for example, if you're thinking about clinical trials, when you start a clinical trial, so the screening visit or baseline visit is a time point where clinical trial starts for this particular individual subject. In this case, it's a snapshot of what you have, versus when you're running a lengthy observational study. So it's not a snapshot, but rather it's a collection of longitudinal information. In this case, certain measures do not make any sense. So also think international. So if you're collecting labs or collecting weights or vital signs and so on, so think about units, for example, right? So pounds versus kilos. So make sure that when you put those units on your forms, they're not just labels, but rather real values. In this case, you will be able to harmonize and recalculate units to the common denominator. So, and then also remember that a lot of instruments, especially questionnaires and function rating scales, have to be validated in multiple languages and even in multiple countries. So, for the same language. So make sure that you do not just take it off the shelf. So another important point to make is that a lot of teams and a lot of ATC systems claim that they're compliant with all this Part 11 and GCP and so on. So, and if they do this, challenge them. The reason I'm saying is it's not just the system, but also the standard operating procedures that those teams have or do not have, and trying to enforce upon you. For example, so Part 11 compliance says that the system has to have unique password and username, which is fine. But if you keep your password on a yellow sticker, in your desk, that invalidates everything. And the first auditor that discovers this will invalidate the entire study. So also when you're putting together common data elements, remember about copyrights, because there are a lot of copyrighted common data outcome measures. And you want to at least to mention those copyright statements, and at most some of them are actually quite expensive, and you have to consider budgeting for those copyrightable tools and utilization. So, and also, if you're looking at this common data elements package, even those downloadable from the NINDS website, there are so many similar tools that mean almost the same, but they're not. So I just picked one from the ALS. So there are 5 recommended depression scales for ALS research. So in a single package. So meaning that you have to, when you're developing your protocol, you have to study what's the difference and make sure that you know which ones you're planning to use. So, and that's kind of the last piece of advice, if you don't mind me saying this, but there are a lot of people who know a lot of things. So people love to share their knowledge, especially if it's free, right? So make sure that you talk to the experts or ask them for help, advice, or references to more experts. This always helps. On this, I'm closing my eyes. And please don't hesitate to contact me. Joy?
Thank you very much to everyone. Do we have any questions? You can use the chat box or unmute yourself on the right.
So one thing we discussed is how do we add new diseases? What is the process for adding? Might be working on a disease that's not covered currently.
Mm-hmm. So we have a new process in place to nominate new diseases. So I think the best way is to contact me and I will walk you through, but the way it is, we're doing an RFI and telling the neuroscience community, this is the form, if you're interested in us to develop a disease, this is the form that you have to complete.
So the RFI is the request for information. How will they know about it when it comes out? Can we send it through CTMC?
Yes, I will. Once it's published, I will make sure that we disseminate it to everyone we have in our listserv. And yes, CTMC is one of them.
Okay, great. And a question for Alex is, have you published results of some of this big data? And if not, what are your plans?
Great questions. Thank you, Robin. So yeah, we did publish certain results, and there are about 30 publications based on that PRO-ACT data set, the one that I mentioned coming from clinical trials. And even after us putting it in 2015, probably 10 to 15 new abstracts are coming from it annually. So there are individual studies that are coming from those multi-site clinical projects as well. And actually, we have the first ever tool that allowed us to merge data in natural histories from two different studies in the same mutation, C9orf72. One is coming from multi-site studies led by Tim Miller, and one is coming from the NIH. So, and because we put together data sharing agreements, data use agreements at the same ICF language and the same actually NeuroGUI, its global unique identifiers, we managed to merge this. Robin?
A question for the audience. How would you all like to get involved with this process? What would be most useful to you? Would anyone be interested in serving on any of the committees for the CDEs, for example? Please send us your information.
I have a question. Can you all hear me?
Yes.
Yeah.
So I'm Will Moyer, one of the course directors. One of the overarching themes in particularly confirmatory clinical trials and all clinical trials in general is that we might be collecting too much information in terms of case report forms and number of secondary outcomes and so forth, and participant burden. To what degree is there consideration for some, I guess, sort of secondary grading of elements? Like these are really high priority to include, you really need to include these for a clinical trial, versus if you are going to, if your design or treatment needs some of these additional scales and secondary analyses, these may be helpful. Because as I put together a early phase study in TBI, I was very impressed by the overall breadth of the number of potential outcome measures I could select from in the CDE portfolio. And as a result, for my exploratory trial, and I know this will come as a shock to many of you, I put in way too many, and it was a little bit. I mean, we're getting the main things, but in retrospect, I learned a lot from the experience. But I wonder how we can help other people kind of learn from those experiences, and sort of if there are ways to kind of upvote particular types of outcome measures or instruments.
Yeah.
I can answer you, Alex. Okay, I can answer you. NINDS, CDEs, and it's similar in other common data elements. So we do have a way to tell people these are the common data elements that you should collect, and these are the ones that are just optional or just pick from the list. So the way we do it is we have a general disease score, and that's general. Like all the neuroscience trials should collect medical history, demographics, and so on. And then there's a disease score that is, for example, for stroke. So this is the list that you have to collect whenever you do a stroke trial. Then we have the supplemental highly recommended, that this is a list that it's very nice that you collect, however, is not mandatory because we don't think that it will fit all the stroke trials. And then there's another one that is supplemental, that is less important, and then the exploratory, that's completely up to the investigator.
Yeah, well, a little bit on the practical side. Yeah, I apologize, Carolina, for interrupting. So basically, your objective is to finish your trial. So with the primary kind of measures, aims, and goals, right? So, and the problem is certainly the more the merrier, but the more you have, the more you have burden on your patient, right? So, and patients start dropping from the trial if it's too abusive, if you wish. So in this case, you have to balance between your desire and all our common desire to aggregate as much as possible. And we have clinical trials that helping us aggregating, for example, collecting biofluid. as part of so industry-sponsored trials, and they're being asked to collect biofluids, and they do it. But only, and you have to think about it, only if it doesn't jeopardize the ultimate objective for you to finish your study, if it makes sense.
There's a question in the chat box. Do the CDE listings clearly mark which scales are copyrighted, and if so, the cost? Or is there a dive-in required on a variety of external sites? And is there a movement in yearly pruning to favor those that are cost-free?
It is indicated, Joy. So basically, a couple of years ago, the NINDS team did a great job putting copyrightable statements on their CDES. It's there, but as I said, new things are being developed. Carolina?
We do have for each CD, we have what is called a notice of copyright, where it indicates at the beginning. It's a very good document that helped me when I work in my stroke trial. It tells you in the heading, you know, this is a copyrighted element, and it will redirect you. It has a hyperlink, and it will redirect you to that website where you can find out, you know, if it's how much you have to pay, or if it's only that you need permission from the PI or the creator of the tool. It also gives you a description, a purpose, bibliography. It's a complete document that tells you right away. In terms of favoring those instruments that are free, I cannot tell you, you know, for sure. In some diseases, yes, but the experts did not focus in that part, and if they were free or not. They were more towards it was the best instrument to capture that data or not. And yes, we take that in account, and we know that's very expensive.
So make sure when you plan a clinical trial and check for whether you have to pay or not, and then make sure that you budget for this instrument. Otherwise, we've seen several examples where people were putting it all together, and then they came after that. Robin?
And I was just gonna, this is Will, sorry. It's relatively unusual for a instrument with a licensing fee to be a core element, or a core outcome in the CDE set. But yeah, I think you just have to do a little legwork to see if there's a licensing fee and whether there's a serviceable alternative. The sort of most impressive one that you may want to be careful about is the risky medication adherence. And if you search the popular media, there's some interesting stories about that one. So again, I don't know if that's in any of the specific disease states here, but you know, if you're designing a study, think about whether there might be an alternative, or at least do your research in advance for the licensing type agreements.
Yep. Go ahead.
We have about 1 more minute. Are there any other questions, comments from anyone?
So Joy, these slides will be available to the participants?
Yes, so on our webinar page of the CTMC site, I will post the video to this webinar probably by Wednesday or Thursday, and I will post the slides with them as well. Okay, if we don't have any other comments or questions, thank you again to our presenters today. I really appreciate the time and your presentation. For everyone who attended this webinar, please evaluate the webinar. The link is in the chat box. And thank you very much. So I appreciate this. Have a good day.